Recently, several moves were made by large AI firms, specifically in the domain of life science research. Google DeepMind published a paper for a model called AlphaGenome in July, and OpenAI posted about this life science research, showing some breakthroughs in protein design using GPT-4o, and also the company's interest in directly applying AI beyond consumer settings and more into scientific research. So I thought this trend that these well-funded AI labs are working directly on science was interesting and a very positive sign to the world of biomedical research in general. And results of the projects themselves were also interesting and are something from which we can speculate on the future trajectories or patterns of how exactly AI, specifically the LLM based AI, can help accelerate bioresearch. So in this video, I want to go over what these projects do, how they connect, and try to interpret the impacts in a broader sense, including what might come next. So let's first start with the big picture. What are some of the ultimate goals of biomedical research, specifically for human healthcare? Well, here's a few: to cure diseases, to extend human lifespan and longevity, and to detect or monitor or diagnose health problems with precision. To achieve these goals, we have to tweak the inner workings of the complex systems of our bodies. And one of the main strategies for doing so is through molecules. So we want to design and create molecules that can lead to a specific effects on the complex systems of our bodies while avoiding other harmful side effects. Now, what kinds of molecules do we want to create? Molecules are made of atoms. There are many different types of atoms, and those atoms can be connected in many ways. So the design space of molecules is very large. It turns out evolution, through billions of years of trial and error, arrived at two fundamental systems or building blocks to make life's work: DNA and proteins. So all the possible molecules out there, at least we have these two to focus on. Well, we often hear that DNA is the information store of our bodies. What does it mean? So when we say it stores information, we mean it literally contains the instructions for building and operating living systems. In fact, people are even exploring DNA as a digital storage medium outside of biology due to the unique characteristics of DNA as a storage system. For example, it is very, very dense and it can store a large amount of data in microscopic scale. A gram of DNA can basically hold hundreds of petabytes of data in theory, and that's like storing most of the public internet in your backpack. And it is also very stable, and it can survive thousands of years if kept in the right conditions. But the implementation of such storage system is still very experimental, but you get a point. Now, DNA by itself doesn't do anything. It just holds information of instructions on how our body system should function. Proteins, on the other hand, are the molecules that actually execute those instructions. They are the molecules that actually do the work. So one way to think of this is as an information querying system. There's a special types of proteins called transcription factors. It's a special type of proteins that can be thought of as a querying mechanism to the DNA. The transcription factors, they ask DNA for instructions on body functions, and the proteins of many different types are then created accordingly to carry out those functions. For example, some proteins act as enzymes. They can speed up chemical reactions in your bodies that would otherwise be too slow to work properly. There are also hormones that travel through the blood to regulate metabolism, and also antibodies which can recognize and kill viruses and bacteria. These are all proteins, and the point is that they cover almost every job you can possibly imagine inside a living system. Together, DNA and proteins efficiently collaborate, forming a loop of information processing to manage information flow and the functioning that basically makes life work. fascinating stuff. And yes, DNA and proteins are the two keywords that you repeatedly see from the two research projects the two big AI labs recently published. If we can deeply understand how they behave, and better yet, design new versions with desired effects, we get closer to those ultimate goals like curing diseases, extending lifespan, and personalizing healthcare. So back to Google's AlphaGenome. You might have heard of AlphaFold before. Well, AlphaGenome is a new model that came out this July 2025. As you can tell from the names, both are related to bioresearch but focus on different problems. AlphaFold focuses on proteins. The model predicts just the shape of a protein sequence. AlphaGenome, on the other hand, focuses on DNA and predicts a variety of traits given a DNA sequence. Both models are called predictive models, with the goal to predict certain traits given a sequence of biomedical data. And yes, the two together cover exactly the two main building blocks of life. So unlike AlphaFold, which focuses on just one characteristic of a fixed protein sequence, its shape in 3D structure, AlphaGenome asks two questions. What does a segment of DNA do, and what happens if we change a single letter on that segment? As it is written here, AlphaGenome is a model that advances regulatory variant effect predictions. What does it mean by regulatory variant effects? Well, small variations in a genome's DNA sequence can alter an organism's response to its environment or its susceptibility to diseases. So basically, a single letter change on DNA can change how a gene is regulated. For example, whether it is turned on or off, in which tissue, etc. But deciphering how the gene's instructions are read at molecular level and what happens when a small DNA variation occurs is still one of biology's greatest mysteries. This is the problem that the AlphaGenome is exactly targeting. So let's start with the input and output of the model. AlphaGenome model is a sequence to function model. The input to the model is DNA sequence, and the output is a list of functions. Given a very long DNA sequence, AlphaGenome predicts experimental signals along the sequence called tracks. These are basically huge arrays describing various epigenetic signals across DNA. Besides just predicting these signals given a fixed DNA sequence, we also care a lot about variant effects. So if I flip a letter from A to G on the DNA, how does the predicted signal change? And how does it change locally or more globally? Now, models for DNA sequence functions are new, but there are several features that make AlphaGenome special. First is long context. AlphaGenome can handle DNA windows up to 1 million base pairs. And that is important because regulatory elements can act from hundreds of thousands of bases away from the gene they control. Most previous models cannot process sequences this long. And similar to conventional LLMs for chatbot AI, processing long input is a significant technical challenge because there's a trade-off between capturing long-range interactions versus maintaining the predictive performance. Google is able to achieve this by building the model on their computing infrastructure, which are mostly these processing units called TPUs. As written here, the largest model was trained in parallel across 8 different TPU devices. And the other feature of AlphaGenome is the multimodal outputs. Basically, the model can predict signals at the level of single nucleotides while also predicting across many different biological readouts or modalities. So these characteristics touch on the trends that we've seen on conventional LLMs. Large unifying models with multiple modalities with long context dominate. The multimodal output obviously can give you a more comprehensive understanding of a DNA sequence, and by learning to predict multiple modalities simultaneously, the model is forced to learn complex interactions between those modalities, which can improve predictions overall. The first step they did in this research is to create a large model trained by 8 parallel computing devices. And next, they also had this distillation step, which means they turned that super large model into a smaller one that can be run on a single device. And that is only possible when you already have a very strong large model from which you can distill the knowledge into this so-called student model. Well, the current version of AlphaGenome obviously has a lot of limitations, but with this model, in general, it is much easier and faster for scientists to tell. For example, if a certain letter in a sequence is changed, it will affect splicing, which can increase the risk for a certain disease. The current model is not yet ready for personal use, but imagine if you have your DNA test results with you with some letter changes or SNPs representing variations in your DNA. And the DNA tests usually just say this SNP is associated with obesity or something, which is very broad and doesn't explain mechanisms behind the association. But in the future, models like AlphaGenome might be able to explain such association in more details, explain exactly what's happening, what are the pathways, and so on. Again, AlphaGenome focuses on one of the main building blocks for life, DNA, and on the other building block, proteins. Recall that AlphaFold is a protein model. It is a predictive model. You give the model a protein sequence, a string of amino acids. The model predicts a key trait of the protein, its shape in 3D structure. Now, predicting structure is one thing, but where do those inputs sequence come from? It turns out that designing a useful new protein from scratch is another big topic in biology, and it is actually one of the hardest tasks. And this is exactly what the new OpenAI research on life science is about. Unlike predictive models like AlphaFold and AlphaGenome, it is a generative model. Yes, the same generative model you use on ChatGPT. Well, why do we want to do that? Why do we want to use a generative model? Again, the design space of proteins is just so large. So as a result, sequences that actually function at all are extremely sparse in the huge random space of all possible proteins out there. That is why generative AI is a good fit. Instead of guessing blindly, a generative model learns patterns from natural proteins and related data. It can then propose sequences that, for example, are more likely to fold, to remain stable, and to achieve some other desired biochemical functions. This reduces the search space from impossible to something more manageable. Again, machine learning applications in protein research existed before. Even the use of generative models and specifically language models is not new. So, is there anything particularly interesting about this approach adopted by OpenAI? What is new is that instead of training a protein specific model from scratch, OpenAI fine tuned a large general purpose language model, the ones used to answer your questions on ChatGPT. And that model already encodes world knowledge, including biology and chemistry. So it starts from a very rich base of knowledge. Then they fine tuned and distilled the model on protein specific datasets. This may include sequences, functional annotations, some other type of structured data, or just text data. This multimodal fine tuning lets the model connect dots between sequences with raw data on structures, functions, and even just scientific text. So, what are the benefits of doing so? Again, large unifying models with multiple modalities win. The model starts from the state already understanding the world, including specifically biology, biochemistry, etc. And from the start, it already kind of gets it. Now comes the fine tuning and the distillation steps. And this is also a multimodal process. The input is not only protein sequences, but many other relevant data formats. Now, there are so many different types of proteins to design, and this project focuses specifically on transcription factors. This is where it links back to the previous discussion on genomes and DNA sequences. Transcription factors are proteins that bind DNA and control which genes are turned on or off. They are one of the most important regulators of cell identities. In this project, the focus was on the famous Yamanaka factors, which are four transcription factors that can reprogram ordinary cells into reduced pluripotent stem cells. And this is basically a foundation of regenerative medicine. But the existing wild-type transcription factors are still too inefficient and risky to be used in practice. So what we want is to be able to come up with variants of such factors that are actually effective and risk-free. And the main task of OpenAI's fine-tuned model, GPT-4b Micro, is to propose such new variants. Interestingly, the AI-designed proteins are not like the ones created by humans, which are usually just small mutations, small number of edits. On average, OpenAI's model generated protein sequences that differed from the originals by more than 100 amino acids, and yet the sequence can still fold and function correctly. That's the level of progression that would be nearly impossible to create by humans alone. So how it works is like this. First, the scientists prompted the OpenAI model GPT-4b Micro to generate sequences with specific desired properties. Well, they didn't disclose how exactly the prompt and the input looked like, but you can imagine it to follow a certain structured format like JSON and has a mixture of technical specs and natural language text specifying the desired protein properties. You have the generated sequences, and Retro, which is a biotech startup OpenAI partnered with, built a web lab screening platform to test those sequences. They tested generation of two types of transcription factors, and both showed a significant increase in the hit rate, meaning that a large percentage of the AI-generated sequences are superior to the wild-type ones in terms of pluripotency markers, even when the proposed models are deeply edited, differing by more than 100 amino acids. Again, such deep editing is difficult to be done by a human due to the large search space. So, in summary, results from this project showed several promising aspects applying AI to bio research. There are several direct effects. We now have methods for faster and more efficient reprogramming of cells. And from there, we have the chance to be able to produce iPSC stem cells at scale, which can allow us to treat various diseases. Specifically, we can target aging hallmarks, as described in the report. The enhanced factors can more effectively reduce DNA damage. There's obviously more to how aging works, but it certainly showed a potential path to improve cell rejuvenations and improve ways for cell therapies. And here are some of the broader effects of such research, which, by the way, are similar to those demonstrated by the AlphaGenome research as well. So it is a proof of concept of the effectiveness of general multimodal models applied in understanding and designing molecules. And the model was not designed solely for Yamanaka factors or longevity. It was built for the general use case of protein engineering and can be applied to redesigning other types of proteins, even other types of molecules in general. So that's it for the two research projects. While DeepMind's AlphaGenome is learning how DNA controls gene activities with instructions or commands coded with the four-letter AGCT, OpenAI's model is designing the proteins that can query and execute those commands. Now, results and the experiments from these two projects are still quite narrow. The tasks are still quite specific, but you can kind of see how things are going and how these projects are starting to connect, especially when you also consider other previous efforts like AlphaFold. So let's zoom out a little bit and look at a broader picture of how AI is helping biomedical research. From both research projects, we observe the same strategy. We start with a very large foundational model, and then we fine-tune the model and distill into specialized versions. Looking at the past few years, the success of general-purpose LLMs, if there's one takeaway, it is that this strategy, this pattern of training models, works magically. You first train model with a tremendous amount of data with extreme level of diversity of tasks and domains so that the model reaches a state where it gets how the world works in general by connecting dots from the training data and building an internal world model. Once the model has a general understanding of the world, things are much easier from there. You then specialize the large model for practical applications at lower cost and/or better specialized performance with well-established machine learning techniques like distillation and the fine-tuning. Note that specialization and distillation here can be on anything. It can be on the model size, which allows you to lower the cost, and also can be on the broadness of knowledge domains, variety of tasks, modalities, and so on. Such a pattern of training AI models is key to why major providers like OpenAI and Google were able to lower the costs for their LLM models so sharply while still maintaining the capabilities. And these well-funded AI labs are now demonstrating that the same strategy works in advanced biological research as well. So, what I find most exciting about this pattern is that it doesn't stop there. You have the specialized lower cost model, and that is where you start to enter a self improving loop. Once data is collected from the successful real life application of the specialized models, we can loop back again to create better large unifying models with more data, more modalities, etc. And again, that will allow us to distill even better specialized models, and the loop continues. So here's a more concrete blueprint. We start with building super general models, all language, visual, audio data, all domains. We then use the model to distill broad biology models that can do all kinds of biological research tasks, unifying tasks like prediction and the generation. We then further distill the model into more specialized model with specialized domains, modalities, tasks, following specific research needs. We then acquire high quality data from applying such models. Both direct model output and interaction data derived from lab tests are high quality experimental data we can use for further training. We then use that data to train the next generation of large multimodal models. This creates a feedback cycle. Each round of application produces data that improves the next round of models. Over time, the loop fills in our gap for understanding and controlling the complex systems of life, of our bodies, not just DNA and proteins, but everything beyond. So, to quickly wrap it up, from the two research projects, we first see some tangible impacts on public health in general, showing that we are on the right track to achieving more precise genetic diagnostics, safer and more effective regenerative therapies for longevity, and so on. We've also gained some insights on the bigger AI pattern of this cycle between giant foundational models and specialized smaller models. That's like the playbook I think will be driving biology research forward in the near future. And I'll be keeping an eye on new progress at this, so if you want to stay updated, make sure to subscribe for future videos.